Jai Hind everyone. Here we are going to continue our class for the transcription. Yesterday we studied about the transcription, so I will revise it again. So DNA transcription is just forming of the RNA from the DNA. That is a transcription. That RNA can be formed from the RNA in case of virus only. Okay. So the prokaryotic or eukaryotic cells, the transcription will be done from DNA to RNA, and it is depend upon an enzyme which is called DNA dependent RNA polymerase. The features of the transcriptions are that it will require the NTPs, ribonucleotide triphosphates. The transcription will be always unidirection, means it will go from one end to the different end, but not in the both direction. Okay, as in the replication is a bidirectional. So remember it, transcription is a unidirectional. So in replication we required a primer, but in transcription we don't require any type of primer. The transcription is a primer independent. And the third and the fourth point is that it occurs only on one strand. It will not occur on the both strand, but replication is occurring on the both strand. So I told you here one point. The DNA is a compact, and every enzyme is not required for the every time. Okay. So if I will tell you the enzymes which required after a maturity, in case of female like estrogen, progesterone, these hormones are increased suddenly. Okay, after a puberty. Okay, so before puberty the estrogen in hormone is not producing. It means the part of the DNA which is producing the estrogen, it can be folded compactly, so the enzyme will be not produced. Okay, so that part which is not transcribing, or the part of the DNA which is not transcribing, that is called heterochromatin. And the part of the DNA which is transcribing, you can see here the part of the DNA which is transcribing, it will be very simple and it is open. Okay, so it is a one thread like structure here. Here it is a one thread like structure. It will form. It will proceed. It will process the transcription. Okay, that region of the DNA is called as a euchromatin. Okay, that region is called euchromatin, can be transcribed. After that, I said the DNA transcription can be different in the it is a difference in the prokaryote and different in the eukaryotes. So in case of the prokaryote, the transcription can be like this. One promoter gene will produce so many type of enzymes like beta galactosidase, permease, transcription, transacetylase. These all will be produced from the one promoter, and it means here the one gene is producing this many three enzymes, three proteins it is producing. Okay, so that is called a polycystronic transcription. So here I showed that one gene will produce more than one protein. That is polycystronic. One more thing is that in one only one RNA polymerase is required to synthesize all the RNAs. Okay, this RNA polymerase required only one type of RNA polymerase will be there, which will synthesize all kind of the RNA. But in case of eukaryotic, if you will see beta, in case of eukaryotic, it is called one gene one protein hypothesis. Okay, like one gene will give one protein only. It is given by and totem. Okay, that scientist name is this Bed Bedell and Totem. Sorry, Beadle. Bedell and Totem. So Bedell and Totem give this one gene, one protein hypothesis, and that type of transcription is called what? Monocystonic. Okay. So in case of eukaryote, RNA polymerase are of the three types: RNA polymerase 1, 2, and 3. And RNA polymerase 1 will produce rRNA, majorly rRNA. Along with that, it will produce small 5.8 srRNA, 18 srRNA, and 28 srRNA. In second, it is producing RNA. The RNA polymerase 2 will produce mRNA, miRNA, snRNA, and lins. Lins is also one RNA. And then rRNA is producing, sorry, RNA polymerase 3 is producing tRNA, SIN is SINs, and U6 snRNA. So these are all important things which we have to study. Now the concept of the transcription unit, the portion of the DNA which is going to be transcribed, that is transcription unit. I shown you here one DNA, and I opened one part and I colored this with the purple color. So if you'll see, this is a mm, wait just a second. Okay, here this is a 3 prime. Okay, and here it is a 5 prime. So the upper strand is from 3 prime to 5 prime. This is the 5 prime, this is the upper strand, and the lower strand is from the 5 prime to 3 prime. Okay, so whenever you are seeing 5 prime to 3 prime, you have to see from one origin only, don't see from the other points. So I seen from this point. Okay, the upper strand which is on which the DNA will be when on which the RNA will be formed, that is called the template DNA, template strand. Okay, so this is template strand. So the 3 prime to 5 prime is called as a template strand, and this template strand has the different name, that is called non-coding negative strand or nonsense strand. The other strand which is called which is from the 5 prime to 3 prime, that is called the non-template strand, and this non-template strand is also known as coding instant, positive instant, and sense instant. Don't confuse with that. Coding instant will give the RNA. The coding instant will not give the RNA. Okay, the non-coding instant will give the RNA. So why we are calling it as a coding instant? Because it is a misnomer. We can say we cannot satisfy this name. So this is a misnomer kind of thing. You just have to remember the template strand will give the RNA only. Okay, and the coding strand is also known as a positive strand and sense strand. Again, I am repeating because you will confuse again. So on the 3 prime to 5 prime strand is my template strand. The template strand is also known as a coding strand, negative strand, or nonsense strand. These non-coding strand, negative strand, and nonsense are the misnomer. Okay, actually this will be giving the RNA. Now come to the different strand. Okay, other strand. The 5 prime to 3 prime strand will be called as a non-template, on which the RNA never going to be produced. On this strand, the RNA will be not forming. So this is called as a non-template. Now the its other name is coding strand, positive strand, and sense strand. So remember it with the template strand and non-template strand. It is easy to remember. The site from where the DNA transcription started from, suppose this is the site from where the DNA transcription started. Okay, so this site is where DNA transcription started. Before this, the DNA will be there, that is called the upstream, and the after that DNA will be there, that is called the downstream. So the upstream will be the negative, I written here, and the downstream will be the positive, I written here. Okay, so what is positive and negative? We are saying positive and negative in the sense of nucleotide counts. Okay, so whatever the nucleotide will be before this transcription initiation site, that is called the upstream site, and after that, that is called the downstream will be the positive counting nucleotide, and upstream will be the negative DNA nucleotide countings. Okay, so I will tell you that how it will called as a upstream and negative. Till now we studied like this, and I shown you there is a one RNA. The priming of the RNA will be the 5 prime to 3 prime. Okay, the new RNA will be forming from 5 prime to 3 prime. Hope so till now you understood all these things. Now we are going to continue the class. Let me check whether the recording is going on. Recording is going on. Then, so the today we will see the three steps of the DNA transcription. First step will be the initiation step. It will be the initiation initiation steps. Okay. So initiation step means what? First, the DNA is a very long again. Then, whenever you want to find the exact position from where you have to start the transcription, you required what you required, better. You will require some sequence, DNA sequence, the particular sequence to find our transcription start site. So the promoter I told you there in the example above that. So the promoter is what? The promoter is having a type of important thing that is called sequence. Consensus sequence will be there. Okay. So the consensus sequence will be differ from the gene to gene, or it will differ from the organism to organism. So the consensus sequence is what? It is the consensus sequence. Consensus sequence is the sequence which will be repeatedly coming again and again. So there is a six base pair consensus sequence. Okay. Suppose I will write here. I will erase it again here. Let me give me just give me 1 minute now. So the if you are seeing online, you can fast forward it. That's not a problem, a big deal for you. Okay. Now, so the consensus sequence I said, the consensus sequence will be the what? The repeated sequence. Okay. Consensus sequence. This will be the repeated sequence. Okay. We also seen this type of sequence, that repeated sequence in the replication. Now the consensus sequence will be the repeated sequence. If you'll see the this type of sequence, suppose mm, A T T A T A. So this will be the it is A T A T T. Wait, A. So this is the consensus sequence. It will be repeated again and again, and that sequence is started from the minus 35. Suppose this is a prime no. If it is my if this region is my promoter, sometime it is not working, huh? So if this is my promoter, okay, this is my promoter region, and if it is a promoter region, before that will be what I told, that it will be negative. So this is my upstream, and this upstream region is called negative. Okay, so if you go upstream to 35 nucleotide, and there you will get the some important sequence which is called consensus sequence. After that, you will get the at the minus 10 position. Okay, at the minus 10, if you'll go from the promoter, this is my promoter. If you'll go back from here, if you'll go back from here minus 10 nucleotide, you will get this sequence. Okay, this sequence will be called as a pribno box. What it is called? Pribno box. Now this pribno box will be recognized by one factor of the RNA polymerase, that is called sigma factor. Okay. Okay, sigma factor is having a big I. Okay, this big I is searching for what? This big I is searching for this pribno box. Whenever it will see this pribno box, it will allow it have a small tail. Okay, I am giving you the structure. So this is a sigma, and this sigma have the small I which will search, which is searching for the what? Which is searching for the pribno box. If the pribno box will be found, then the sigma factor will allow the RNA polymerase to bind here. Where it will bind? There is a two strand, above strand and the down strand. Okay, so it will bind to the above strand because it have to be transcribed that portion only. So we are seeing what? Initiation. Okay, remember it. So initiation is done. Now our RNA polymerase will bind here. So I will show you how it will bind. Suppose this is my RNA polymerase, and it is binding here. Okay. Okay, this RNA polymerase will bind here, and finally, once the RNA polymerase will be bind, it will start. It will start adding. It will start adding new nucleotide. Okay, it will start adding new nucleotide. So this will be the RNA polymerase, and it will start adding new nucleotide. So why I added above, because this is my 3 prime to 5 prime. Okay, this is my the strand which is 3 prime to 5 prime. Okay, so here the sigma factor will be there. It will be here also. First is will search, and then it will help in the initiation. So this will be the sigma factor. Okay, did I made okay. Okay, this is my sigma factor. Now sigma factor, once the initiation will be occur, remember the initiation will be occur on the template strand only, the 3 prime to 5 prime. Once this initiation will be occur, the sigma factor will be detached from here. It will detach, and then finally it will start the elongation. Now initiation is done. Okay, now we'll see the elongation. What we are seeing in this, we are seeing this transcription in the E. coli, or we can say in the prokaryotes. In eukaryotes, almost it will be similar. I will tell you what is the difference in the eukaryotes. So in case of the elongation, you will see the. I will copy this diagram again. Mhm. In case of elongation, the RNA strand will start forming. Okay, so this is my 3 prime. This is my 5 prime. This is the opposite strand. This is from 5 prime to 3 prime. And now this sigma factor which you seen beta, that sigma factor will be not going to be here. It will be now detached from the. Okay, now this will be detaching from the. Okay, got it. Now this sigma factor will be detached from here, and it will start binding. It will start binding the new nucleotide. So this is the here the RNTP will come, the ribonucleotide triphosphate. So suppose this is the first ribonucleotide triphosphate, then it will require. I have to make it with a different color. So new nucleotide is coming here, and it will be attached like this, and it will start it from here. Okay. So this will be the what? This will be the 5 prime, and this will be the 3 prime. As it will move forward, it will start forming a complete chain. So this is the elongation only. It will move forward, and the strand, RNA strand, will be started forming. In case of this in case of the E. coli, the translation will be begins with the transcription itself. Okay. So here you will see the ribosome will come, and this ribosome will come and start forming the protein. Okay, here new protein will be formed. Here protein is start forming. Okay, so in case of this is ribosome. In case of prokaryotes, the polysome you will see. So many ribosome is coming here. More than one or more than two ribosomes will come started coming here. Then it will called. Then it is called beta polysome. Okay. And this can be possible in the prokaryotics only. In eukaryote, once the transcription, until the transcription will be not completed, the translation will be not occur. So in case of here, you can write in prokaryotes, in case of prokaryotes, transcription and translation are occurring together. Okay, prokaryote, transcription and translation occur simultaneously. Okay, like together it will occur. But in case of eukaryote, it will not occur together. So hope so. Till now you understood what is the thing.
Can you explain about elongation again?
Okay. So elongation is the simple thing. It doesn't have to do too much of thing. What I was saying, the elongation will once the elongation will start once the initiation will be finished. The sigma factor will be released. Okay, this is the sigma factor here. Sigma factor of the RNA polymerase will released. Okay, its work is done. Now it will removed, and then this is a RNA polymerase. What it is? RNA polymerase. This RNA polymerase will start adding the ribonucleotide triphosphate. Okay, so slowly it will add so many ribonucleotide, and it will form the RNA from 5 prime to 3 prime. So I told, in case of prokaryotic, what it is happening, the as the RNA is forming, the ribosome start binding here. Okay, and it will give the new protein. It will give proteins. Okay, it will give starting formation of the translation. You can say directly translation begin. Here the transcription is not ended, but the translation begin. So that is called that this so many ribosomes once it will attach to the mRNA, then it is called beta polysome. Okay, so polysome will seen in the polysome will seen in the prokaryotes. And I told the prokaryotes will do the transcription and translation together. Okay, that I told, and hope you understood this. The problem is that in case of prokaryotes or in case of other eukaryotes also, we can see that RNA can be form a supercoiling. Okay, if it will be there, then it can start forming like this. That will be the difficult situation. For removing this, we are putting some enzymes which is called the gyrase enzyme. What we are putting? Some enzymes. Okay, we will put some enzymes. So once this enzyme will be put down, the strand will become straight. Okay, the strand will be become straight. So this is the topoisomerase enzyme, or we can say gyrase enzyme. This gyrase enzyme already we seen in the replication. Okay, this is gyrase enzyme. Okay, it will remove positive supercoiling. Okay, got it. So I gave you diagram here only. The diagram which I shown you here, it should be shown here at the end, but again I have to erase it. So I showed you here that how that coiling will be occur. Positive supercoiling can be occur, and it will be removed by the gyrase enzyme. Hope so. This handwriting is visible or understood. I will. It is not too much good, so I am writing it again. Gyrase. Okay. gyrase enzyme. So till now you understood.
Yes,
Okay, now move to the next step. So that this is the time to terminate the transcription. So the last will be the termination. So the termination will be done by what? Terminator. There will be one terminator. The terminator will be root side. Okay. So termination can be done in the two manners. I will tell you here, and this is important thing. You just see here. Okay, like focused here. I need your whole attention here because it is important. It is going to be important point. Okay. Suppose from here to here, the our RNA polymerase came. Okay, it is putting the nucleotide, ribonucleotide. Okay, and uh, this is our RNA. Okay, this is our RNA. The priming will be same. The it will be the 5 prime, and here it will be the 3 prime. Now it have to be stopped. It have to be stopped. So how it will be stopped? It will stop like that. There will be termination can be of two types. First, I am going to tell you intrinsic terminator. Okay, it is of two type. I am going to tell you intrinsic termination. In intrinsic termination, what is happening, beta? That there will be one sequence which is very rich in the G and C. Okay, there is a sequence which is very rich in G and C. Suppose G, 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 then again C, 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 like that. Okay, or here you can take T. Like that, it will be forming. Okay. So if this RNA, suppose if you are going to make this RNA, so what it will become here, beta? This will come. This will come C, 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 A, G, 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 G. Up to here, you don't have any problem. Okay. But it will go, if it will go further, it will form a stem loop like structure. Okay. This is the next diagram I am showing you here. If it will go further, it will form a stem like loop structure. So here it will be C, 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 C. Here it will be G, G, G. Okay. So again, one more G. So it will form this stem loop like structure. Once it will be forming the stem loop like structure, it will stop. Okay, it will the DNA polymerase. The sorry, DNA polymerase. I am saying the RNA polymerase will be stopping here, and it will form a hairpin like structure. Because of forming of this hairpin like structure, because of forming of this is hairpin structure, the termination occur. Okay, then termination occur. Got it. Other another method is what? Another method is raw dependent. Oh, that is a raw dependent. Raw is a one factor. Raw dependent termination. If you will see this raw dependent termination, this RNA polymerase which is there already we seen. I can make a copy from here. In raw dependent, what you will see, beta, the RNA polymerase is there, and it have the priming of from the this is my template DNA. So the template DNA have the priming 3 prime to 5 prime, and here you will find there will be one There will be one enzyme which is called raw enzyme. Okay, okay. This will called as a raw factor. Okay, this is a raw factor. Once the raw factor will bind here, then it will terminate. It will terminate here. Okay, there will be okay. There once the raw will added RNA polymerase, the transcription will be the transcription will stopped or finished. Okay, so you will get the new RNA here, and it will fall off, and its priming will be 5 prime to 3 prime. Here you will have the 3 phosphorus, and how you will get this 3 phosphorus, that will be the different story, but you just remember there will be one 3 phosphorus, and it will be removed till now. And I think don't have any doubt, and you just write the antibiotics for this transcription. You seen the antibiotics for the DNA also. Now we'll see the antibiotics or the inhibitor. You can say inhibitor for the transcription. Inhibitor for transcription. If you'll see the inhibitor of the transcription, the inhibitor of the transcription are the rifamycin. Okay, rifamycin. I don't think it is required to tell how it will be blocking in for your level, so I'm not telling you here. And the actinomycin D, actinomycin. These are the transcription inhibitor. They will most of the time they will ask this type of question: what is the inhibitor for the replication and the transcription? Okay. So now we have to add some points. We already added some point for the eukaryote. Just remember this polysome or this transcription translation will be together in case of the eukaryote. Sorry, in case of the prokaryotes only, but in case of the eukaryotes, that will be not together. Okay. Now the in transcription you can add instead of the privno box there will be one tata box. Okay. Instead of privno box there will be one tata box for the transcription in the eukaryote. The important thing is there in the eukaryotes. The we have to what we can say, we have to, the word is not coming in my mind. Okay, we have to convert the hnRNA into mRNA, or the RNA will be uh, the word is not coming. The it will be regulated, no? Okay, the RNA will be modified by the addition of the 5 methyl 5 prime methyl at the 5th prime, and poly A at the 3 prime. So that is called modification of this mRNA, and that will call it the RNA processing. So RNA processing we'll see here. So RNA processing will be in case of mRNA. We are seeing the RNA processing in the eukaryotics. RNA processing, that is the word which I am searching in my mind. Okay, RNA processing will be occur in the eukaryotes, and the RNA processing will be done by addition of the seven by addition of the by addition of 7 methyl guanosine, okay, at the 5 prime. So this is my RNA which you seen here, and it is fold from the transcription, and this is a 5 prime, and this is a 3 prime. Now if you'll see beta, this is the phosphorus is, you already seen there was a one phosphorus in beginning. Now you will find why it is 3 phosphorus. Now the 5 methyl guanosine will be coming here. Okay, there will be 7 methyl guanosine. Okay, so this 7 methyl guanosine will come here, and it will remove the 2 phosphorus. Okay, then this 2 phosphorus will be removed. Once this 2 phosphorus will be removed, then only you will see the diagram like this. You'll see the diagram like this. Here it will be guanosine, and then one phosphorus. There will be one guanosine, phosphorus, adenosine. There will be one guanosine, phosphorus. That is alpha. Then there will be one beta phosphorus. I have to tell you what is alpha and what is beta phosphorus. Huh? I will tell you later. Okay. So this will be the structure, and this is a 7 methyl. Why it is called in 7 methyl guanosine? At the seventh nitrogen, at the seventh position of the guanosine, it will having the methyl group. Okay, so that is called guanosine methyl. Me. You just write methyl guanosine. So this methyl guanosine will be added here, and why it is required to add it here? The it will okay. First, this is called capping. Okay, what it is called? Capping. So capping means what? Adding of 7 methyl guanosine. Adding of 7 methyl guanosine at the at 5 prime of RNA. Okay, 5 prime of HNRNA. You can say HNRNA. You'll add 5 prime of HNRNA. Okay. Now you added this. Why it is required? The point is that you require this capping because of it can be degraded, so it will protect from the degradation. Okay, the mRNA will be protected. Protection of mRNA from degradation. Okay. Next, what it will do? It will help in binding of ribosome. Third, what it will do? This RNA is formed where? This RNA will be formed in the nucleus only, because in DNA is there in the nucleus. So RNA is forming in the nucleus. Okay. So the RNA should be transported. It will help in the transport of mRNA from nucleus cytoplasm to cytoplasm. Here we will not stop because the processing is required still more work, and that is what? That is the polyadenylation. Okay, so it can be more, but I will tell you. Should I tell you here only? So the more after once the transcription will be finished, the A will be added here. Okay, the poly A. So the A will be added here at the end of the 3 prime. This is a 3 prime and this is a 5 prime. So at the end of the 3 prime, the poly A will be added here. So that is called polyadenylation. Okay, and here the processing will be done. So the two process mRNA is processed by the two manner. First the mR capping, and second is the polyadenylation. Okay, so would like to write it here. And the first was what? First was the capping. Okay, and second was what? Polyadenylation. So the polyadenylation will be occur at the 3 prime end of the mRNA and by the polymerase enzyme only, RNA polymerase enzyme, and that will be the RNA depend the template independent RNA polymerase. This A is added now. So how this A will be added? It will be not according to the DNA, because till now whatever the green you are seeing now from here to here, it was complementary to the DNA. Now this reason is what? It is not complementary to the DNA. It is added. So after the formation of the mRNA, it is adding. So it is done by the polymerase only, but it is done by DNA independent, or you can write template. What is my template? That is my DNA. Okay, DNA nation. Okay, template independent RNA polymerase. What happened? Why it's not writing? RNA polymerase. This is the important thing you just added here. Okay, now this is finished here. The RNA processing. Tomorrow we'll see about the tRNA and rRNA. Among this all RNAs, the rRNA will be the more. It is around 80%. Okay, the rest of the in eukaryotes we are seeing usually the rRNA will be more in, and remaining will be less only. Okay, exact percentage you can no need to see. So later we'll see some processing. More processing will be there in the RNA. I should tell you here only. One more thing I want to tell you: the sequencing. After okay, splicing I will see. I will tell you now, and then I will solve some questions, and then we'll finish it. Okay. So till now, do you have any doubt? You can ask me if you have any doubt. So, my dear students, oh. So till now we seen the methylation. Sorry, not methylation. 7-methyl guanidine addition at the 5 prime and polyadenylation at the 3 prime. That is very important. You just remember it. Now we'll see what I will see the splicing. Okay, splicing will be the third RNA processing process, and it is splicing. Okay. So splicing is the first we see. You will splice something. Splice means just cut the some parts. So here we have to cut this segments. Like this black segment will be there. This black segments is in the or mRNA. Okay. This mRNA segments is not required, and this is not going to form any protein. So this is called intron. What it is called? This will be called as a intron. and that is called, the green part is called exon. So this exon is going to express protein. It will express for protein. Okay. And then this intron will be the interfering. Okay. Interfering segment. Okay, it's not required in the mature mRNA. So if it is not required in the mature mRNA, we have to remove it by the process called splicing. Okay, splice it. So what you will do, beta, you will take this mRNA, and then you will cut this intron from here. Okay, we will cut this intron from here. Finally, okay, finally what you will do, there will be some complex which is called the spliceosome complex. Okay, so it is consists of so many enzymes which you don't have to remember, and no need to now. So the spliceosome will come. Suppose this is a complex enzyme. Okay, got it. So this is a complex enzyme, suppose, and it will having it. What it will do? It will cut from here and then here. Okay, okay, like this. The spliceosome will come here. The spliceosome will come here. Okay, so what it will do? It will cut these sides. So these are what? Spliceosome. So their work is what? Their work is to cut this phosphodiester ester bonds, and the another work is that it have to uh, it have to cut from the both sides, and it have to remove it. Okay. So finally you will see, you will get the this type of structure. You will see this type of structure. Okay, this here you will get this. Okay, this now it was my the length is decreased now because the region is removed. This will form a circular kind of structure, and it will degrade it. Okay, it will go out. Now this will be added. This this points will be added by ligase enzyme. Okay, DNA ligase enzyme. It will be added by the DNA ligase. So there will be one. It require spliceosome require ATP to assembling. Okay, it require ATP for assembling. So for assembling this big enzymes, spliceosome required a ATP, and that is a ATP dependent. Why I written here ATP? To make sure to tell you that this process is a ATP dependent process. So after adding of the DNA ligase, it will be added completely, and now it is ready for the processing. Now this will be ready for the processing, and finally after forming of this all these thing, then it will be go for the translation. So hope so this is clear now, and we'll move to the next point. What is the next thing? We will see the RNA sorry, tRNA. Or before that, we will complete one more thing I have to teach. That was um, telomerase replication. Okay, telomerase replication we will see. So already we read about the replication. Now better we have to see the telomere replication. What is telomere? So the telomere is the end of the DNA, end unit of the DNA. Okay, so suppose DNA is a big length. So if it is a big length, beta, here, if it is a big length, if it is a big length here, so the telomere will be have the potential to protect from the exonuclease. So exonuclease is what? Exonuclease are the enzyme which will cut the end of the DNA. Okay, end of the nucleotides. Okay, so this exonuclease can cut our DNA also. So the telomere will be there to protect it from the DNA. Okay, so the sequence of the telomere is this is the sequence of the telomere T T triple T or T T A triple G. Okay, so this is the sequence of the telomere. Again, I am saying what is the use of the telomere? It will protect our DNA from the exonuclease. Okay, from exonuclease. Got it? Now, whenever the transcription, the replication will be occur now.
What
So,
happened, Pankaj?
oh ho, I didn't start my voice. Okay, sorry. Oh, repetition. Now, the telomere is what? Telomere is the end unit of the DNA. So, the telomere is the end unit of the DNA. So, the DNA cannot be damaged by the exonuclease. Okay. So, it will be protected from the exonuclease. Telomere will help in the protection from the exonuclease. Got it? Now, the sequence of the telomere is the TTA triple G. This is the sequence of the telomere in the 5 prime to 3 prime, and from 3 prime to 5 prime it is the sequence. Okay. So whenever the replication will be occur, there will be one lagging strand, you remember, at the 5 prime to 3 prime. If there will be lagging strand, you required again and again one, you will require this primer. Again and again you will require this primer. So this primer is having a, it will be put after a some length, like after this, after 200 nucleotide, the primer will be can be put. Okay, so but if the DNA is less than 200 meter, like suppose this is the unit, if it is less than 200 meter, 200 nucleotides, so it cannot be added here primer. So what we have to do, we have to extend this region, the small region which is there now. This is the small region, we have to extend it up to here, up to 200 nucleotides, so we can add a primer. Again, the point is that the primer you are adding here, here, so it cannot add new primer nearby it. You cannot add new primer here, so it have to be added somewhere far from the earlier primer. Okay, around 200 nucleotide. So but here the no nucleotide is there, the our small tail will be remaining. Okay, that is a small tail. Okay, that is a telomere. So the point is that if the replication will be occur as like that, the again and again the DNA will be shortened after some time. You are leaving each time you are leaving this much of nucleotide, each time you are leaving this much of nucleotide. So next time you will leave this much of nucleotide. Okay, so next time you will leave more, and finally the DNA will be shortened like this. So it should not be happen. Okay, so preventing from this, we have a enzyme which is called which is called telomerase enzyme. Okay, so telomerase enzyme have the RNA plus protein. Telomerase enzyme have, this is the telomerase enzyme. The telomerase enzyme have the RNA plus protein. This RNA, the protein will having this RNA. Okay, suppose this is the RNA, and this will be the sequence which you are seeing here, G C C. This is the sequence which you are seeing here, and it will extend the DNA strand. Okay, till 200. So earlier it was till here, now it is extended till this point. So if it will be extended till here, then you can add one primer here. So you will add one primer here, which which will help in the extension of the new nucleotide, and it will cover the, this is the telomere section also. Okay, so if you'll add here a primer, if you'll add here a primer, because how you can add the primer, because you extended this region. If you extended this region, so after a 200 nucleotide you can easily add the primer. So if you'll easily add the primer, the this section will be also replicate easily. Got it? So finally you will get the proper length of the DNA. Got it? So this is the DNA polymerase replication. Sorry, this is the telomere replication. So nucleosome, this I didn't mention. I don't want to mention it here. Okay. So this is the telomerase replication. It is done by the telomerase enzyme. This is very important, this telomerase enzyme. If the telomerase enzyme will be not there na beta, so after some time your cell will have the small DNA and loss most of the gene. Okay. And it will be not protected from the exonuclease, so that will be the like difficult or serious condition. Okay. So for extension, we have the telomerase enzyme. Got it? Hope so. It is understood.